cholesterol that we have is synthesized in our bodies, not coming from the diet. But what happens while we transport this VLDL is that we activate a series of enzymes, in particular lipases, that will start to hydrolyze. Once this VLDL will get recognized from the cell membrane of our cells, especially skeletal muscle cells and adipocytes, we start to hydrolyze triacylglycerides, and as we will see later in this today's class, we will metabolize them and use them as an energy source. Once again, once we have free fatty acids and glycerol in this stage, the free fatty acids can be either directly used if we need energy, or they can be stored, and so we can regenerate triacylglycerides inside our cells and stock this excess of lipids. Now, before the break, we were talking about what is called bad and good cholesterol, because we said that the VLDL, once by floating around in our blood and reach the different cells, get smaller and smaller, because the triacylglycerides are taken up by the cells. They get smaller. They change the ratio of the different components, and as we said, there is also a change in the composition of the proteins on the surface. So we form what they're called LDL, low-density lipoproteins. These, are known, even by non-medical people, as the bad cholesterol. And we said that although they're considered bad cholesterol, they have a very important, purpose, because of course they are used to give cholesterol to our cells. So there's an LDL receptor that is able to recognize these lipoproteins, and we can get cholesterol in our cells. But as we said, if we have an excess of triacylglycerides, especially biosynthetic ones from the liver, we can get a lot of LDL in our blood, and if they stay for a long period of time, these are the sources of the atherosclerotic plaque. And the reason is because, of course, these lipoproteins are very rich in cholesterol, and this can really alter the surface of our arteries, especially, as I said, if we on one side do have a metabolic disease, as we will see, where some of these mechanisms are altered, or simply because we are eating too much carbohydrates and we are synthesizing too many of VLDL, and then LDL, will definitely stay longer, because of course we need cholesterol in our cells. But if we have an excess of cholesterol is going around, of course there's an excess of LDL that will not maybe picked up by the liver fast enough, and then they will stay for a longer period of times in our bloodstream, and they might disrupt our vessels. Now, nature knows that we are not always very strict with our diet, but in any case, nature also knows that in our metabolic pathways, we need to get rid of some of the excess cholesterol in our cells. So we designed HDL, high-density lipoproteins, that are, those lipoproteins that are used for picking up the excess of cholesterol from peripheral cells and bring back this cholesterol to the liver. This is where we were at the end of the last hour, and now we will try to understand a little bit more about this reverse transport of cholesterol. Now, as I mentioned already, this is a very complex mechanism. It's actually not fully understood today. So for this course especially, I'm not expecting you to study every single specific pathway, but once again, we need to understand the overall picture and understand where problems may occur. So, of course, if we need to reverse transport cholesterol, we have, on one side, we have to have a specific transporter. So once again, we will have a specific lipoprotein combination that will be recognized by our cells and will activate the reverse transport of cholesterol. And as we said, in this case specifically, we need to have a transporter that at one stage will be recognized by the liver, and this excess cholesterol will be picked up by the liver and then either recycled if we really need this cholesterol, or as we said, we will get rid of this excess cholesterol to the bile, and we will see later on how we do that. So once again, HDL is considered the good cholesterol. When you're going to have your patients, you're going to measure the cholesterol content in their blood, and of course, the higher is the the ratio HDL LDL, the higher is the HDL, the better is for the patient. However, as we saw in the first half of this class, it's not completely true, in the sense that, of course, if we have we induce the production of HDL by, for instance, exercise, this is good for our health. However, attempts to increase HDL with a drug kind of failed, and most likely the reason is because, once again, these processes are very, very tightly regulated, and we cannot really make changes without affecting the rest of the organism. We already talked about the use of statins to reduce the biosynthesis of cholesterol in the liver, as a therapeutic approach for patients that dysregulate the synthesis of cholesterol. Now, let's see how nature designed this transport of cholesterol. Once again, it's a quite complex mechanism, and the overall picture is that we need to remove this excess of cholesterol from these peripheral tissues and deliver it back to the liver. What happens there is either redistributed by reforming the LDL, or it's removed by the gallbladder, and we'll see, with the feces how this thing goes. Now, this reverse transporter of cholesterol, HDL, their synthesis starts with this lipid-poor apolipoprotein A1, which is synthesized in the liver and in the small intestine. In other words, if at the very beginning, we only have a very small protein-rich particle, and basically this particle does not contain, or just contains a tiny bit of cholesterol ester. Now, ApoA1 enters the bloodstream and then start to interact with the cells that it encounters. Actually, this is one of the parts of the mechanism that is not really completely understood. It seems that actually these particles are released by the liver cells and directly interact with the liver cells, and they start to mature by directly interacting with the liver cells. So, in other words, it's sort of like an autocrine mechanism, such as ApoA1 is first secreted and then interacts with the cells that generated to start the nascent HDL. So the nascent AGL, as we saw in the animation before the break, it's a sort of discoidal particle. It's very flat at the beginning, and then while, as we will see later, picks up cholesterol from our cells, it becomes bigger and bigger, and it starts to have a more rounded shape. Of course, this nascent HDL also contain an important enzyme which is called LCAT. LCAT is used to esterify cholesterol. As we said, cholesterol is usually transported as a cholesterol ester, and so we need an enzyme to esterify cholesterol. Now, as you can see from this scheme, basically the nascent HDL is formed by, once again, a quite complex and not completely and not fully understood mechanism, which combines this lipid poor ApoA1 phospholipid that they are found around, also is picking up cholesterol and phosphatidylcholine from the remnants of chylomicrons and the LDL. In other words, there is a sort of a beginning of the formation of this particle. But what is important really is not this part, but is where we start to mature the particle and get and start to interact with cells and do the job, which is extracting and taking away cholesterol from cells that do not need all this cholesterol. Actually, a turning point in understanding this mechanism was finding this enzyme actually is a receptor. It's called ABCA1, that stands for ATP binding cassette subfamily A1. Don't worry, I'm not gonna ask you this kind of question with the exam. There is a receptor that can recognize ApoA1 that is present on the nascent HDL, and this will start this efflux of cholesterol from the cell, and it requires ATP for these purposes, and the end result is that this nascent HDL becomes bigger and bigger because it starts to get cholesterol inside. There are different models that have tried to describe how BCA1 mediates this efflux of phospholipids and cholesterol from the cells to this nascent HDL. You're welcome to look into these. I mean, it's very complex a story, so I'm not going to get into these details, but there are several ways that have been proposed for this efflux. What is important now is that ABCA1 transports this load of cholesterol from inside the cell to the outer surface on the membrane, where ApoA1 picks it up, and this movement is unidirectional. So we are actually only exporting phospholipids and cholesterol out, from the cell to the cell surface. In this process, actually, a key role is played by macrophages, which, as you know, are phagocytes from the immune system, which are specialized in digesting particles, and the interaction with ABCA1 of ApoA1 moves this excess of cholesterol and phospholipids to ApoA1, and this process basically generates the nascent HDL, as you can see that are slightly bigger and not anymore so flat as the beginning. There is another transporter that is important. It's called ABCG1, which actually starts to interact with mature HDL, and this also facilitates the movement of cholesterol out of the cell into the HDL. Also, the mechanism of ABCG1 is still not completely understood. And actually, it seems that it's not that specific, because I have seen that it has been shown that ABCG1 also can export cholesterol to LDL. But anyway, both these processes are critical for exporting cholesterol away from the cells, and especially at the sites where the plaques and the blood vessels are formed, and so high HDL are very important for people that suffer of cardiovascular disease. Now, there are other, non-specific pathways that have been proposed. In total, there are actually 4 pathways. We've seen the first 2. There's a third one, non-specific, of cholesterol efflux from the cell surface. If you think about it, the cholesterol is inside our cell membrane, so there is basically a non-specific exchange between the cell membrane and the extracellular acceptor, mediated by sort of like a passive diffusion, due to a different cholesterol gradient. Another mechanism, fourth mechanism, is again an efflux of cholesterol to mature HDL, but interaction with another receptor, this SRB1, which is called scavenger receptor class B type 1, which again will transport cholesterol to HDL. So no matter how cholesterol gets to HDL, the net result is that, as you can see here, this particle gets bigger and bigger, and finally they are ready for their destiny. We said that they can get back to the liver to be recycled or to be extruded from our body. And as you can see from this slide, this next step can be direct or indirect. So as you can see here, the HDL particle, I don't see the mouse. Where is the mouse? Okay, the HDL particle that can directly go back to the liver or can transfer some of its cholesterol to LDL. There is really two different mechanisms. So either it's recognized directly from the liver, or there is a sort of an exchange of components. So LDL gives HDL some triacylglycerides, and HDL in exchange gives some cholesterol, and this will make the LDL more mature to also get recognized by the liver and picked out by the liver. So, actually, once again, one of the major breakthrough in understanding this complex mechanism of cholesterol return to the liver came actually with the discovery of this ABCA1 receptor. In fact, it was discovered actually in a disease, the Tanger disease, which is a rare genetic disease that is characterized by low plasma HDL and increased cardiovascular disease. So if you have a defective ABCA1, you cannot really form mature HDL particles, and this, of course, will disrupt the possibility of recovering the excess cholesterol, and so these patients do have, an excess of cholesterol ester deposition, especially in the tonsils. One of the way to see if people is affected is the enlargement of their tonsils because of the deposition of these fatty of these cholesterol esters. Of course, this, I'm sure that you will, study this disease more in detail in the future. Now, once, they reach the liver, the cholesterol esters are hydrolyzed, and then the free cholesterol is either converted to bile acids, or, as you remember, it's very important. We produce approximately actually 600 milligrams of bile salts daily, or, as we said, if we really do have an excess of cholesterol, it can be transported into the bile for excretion with feces. So we just throw it away. So once again, this is the physiological condition. Of course, we can have several situations where there are alteration of these metabolic pathways, and we can have disease. So, for instance, there is, you will study another metabolic disease that is called fatty liver disease, also known as hepatic steatosis, which, as you will see, is a condition where basically we have a fat liver. So if you take a look at a slice, as you can see in the picture of your liver, you can actually see fat droplets in the liver. And actually, often at the beginning, especially, there are no or few symptoms. It's typical of, it can be non-alcoholic fatty liver disease because of some dysfunction, or it can be caused by, for instance, an excess of abuse of alcohol, meaning, alcoholism can cause this situation. Once again, if there's an overproduction of triglycerides in the liver, we do not have, we are not able to transport it efficiently. We can actually get stuck with the fat accumulating in so impaired glycosylation, which inhibits lipoprotein release, impaired secretion of the LDL. There are, several factors that can contribute to this disease. Of course, as I mentioned, many times, these lipidemias are diseases that are becoming more and more predominant in the Western world. Of course, some of them have genetic origin, but some of them, of course, can be caused by our lifestyle, and so it's important to control, especially the diet and what we eat. So, for instance, it's very important, this LDL receptor, as you may imagine. As I said, the main problem of hypercholesterolemia is that LDL that contain a lot of cholesterol will stay in our blood for a longer period of time. So, there is enough time, there is enough cholesterol that it can be, that can build up plaques. So, of course, if there is defects in this LDL receptor, of course, our liver is our cells in general cannot pick up, cannot recognize LDL, and of course, this mutation results in a failure to release the LDL cargo and the loss of the receptor by degradation. And so this is also a very, can be the result of genetic, but also of exogenous situations in our life. So, of course, we have, studied all this part together with the other mechanisms that we have seen, our catabolism, to see how we can use food for energy reasons. So now let's switch gear and let's see how, we can use fat. We can use fatty acid for energy. We have seen already, most of it when we studied the Krebs cycle, but as you remember, we entered the Krebs cycle with acetyl-CoA, and so we need to understand how we go from, 3-acylglycerides to acetyl-CoA that can enter the Krebs cycle. Now, of course, as you remember, we already talked about, the hormonal regulation of our metabolic pathways. We've already seen what happens, right, when we are in a fed state or when we are in a fasting state. Now, let's consider how these different states will affect lipid metabolism. As you can see from this graph, of course, when we start a fasting period, after a few hours, within a maximum 24 to 48 hours, we consume completely our storage of glucose. We have, we said already that, of course, our metabolic pathways, they do not stop. They are all working all the time, but of course, there are some that can produce ATP more faster, like, glycolysis. Therefore, when we're fasting, the first thing we can do is to release glucose from, our hepatocytes through glucogenolysis. And as you can see here, the process, of course, needs to slow down while we're fasting because, we consume all the glycogen that we have in our body. So of course, when we are under these circumstances, otherwise we would not, do a diet where we starve ourselves. We know that the end result is that we start to mobilize lipids and we can start to use lipids, right? Today we have discussed this many times. There is this, big debate, right? Shall we activate ketogenesis or not? How much, we should activate it? How can we activate ketogenesis? And we'll try to understand a little bit about this today by understanding how these processes work. So if we, we are consuming our resources of glucose, we really need to use our energy source that is containing fatty acids. As we mentioned many times, to use fatty acid, we need to have two conditions, right? What are these two conditions? We need to have oxygen, right? We cannot use fatty acids if we don't have oxygen because we need to run the Krebs cycle, right? And the oxyphos. And the second? I said it 1 million times, and I always say the students keep forgetting it. I don't know why. We need oxaloacetate, right? So we said it many times that unfortunately we cannot readily synthesize oxaloacetate unless we have sugars, right? We said it many times that we need to synthesize oxaloacetate to enter the Krebs cycle, because in the Krebs cycle we have acetyl-CoA that can directly come, as we will see now, from fatty acids, but we also need oxaloacetate. Now, this oxaloacetate, although we can make it, as we will see, from glycerol, right? So when we hydrolyze 3-acylglycerides, we make 3 fatty acids and 1 molecule of glycerol. We can make oxaloacetate from glycerol. However, that's not enough. So as we already discussed, as we will see, especially under prolonged starvation, what we'll have to do is we will have to use gluconeogenesis from amino acids to synthesize sugar in the liver so that we can actually use fatty acids as fuel. But once again, to run the Krebs cycle, we need both acetyl-CoA and oxaloacetate. Oxaloacetate, as we will see, we cannot really make it from fatty acids. There are some ways to do it. There are some recent reports that have shown that we can actually do it to a certain extent, and I'll show you how. However, if you look in old textbooks, that's what they're going to say, and that's because that's the truth. And that if you're on a diet where you completely remove carbohydrates, it's true that you lose weight, but as we said many times, it's also true that we will lose also muscle mass because we will use branched chain amino acid to synthesize glucose that is needed for the Krebs cycle. Now, of course, if we want to use fatty acid as fuel, we need to be in a situation to use them. So let's consider, for instance, we wake up in the morning, we do not have breakfast, right? We, overnight we used quite a bunch of our storage of glycogen, and so for sure, what is going to be high as an hormone in our bloodstream? Glucagon, right? So insulin will be very low, glucagon is going to be high. And of course, if we are going to go, for a run, or we're going to go exercise, for sure we'll start to produce also adrenaline. So we will have two hormones that will really push us to try to, increase the ATP production, but we will have to do it through our energy, other energy sources. So, as you remember from the first part of the course, the TCA cycle happens inside the mitochondrion. So, if we want to use lipids as an energy source, well, that's quite a complicated and a complex mechanism, right? We need to activate the mechanism. We need to mobilize lipids because, as you know, we have the storage of lipids, especially in our fat cells, adipocytes. So we need to mobilize all these lipids. We need, to hydrolyze triacylglycerides. We need, as we will see, to get them inside the cell that is going to use them. Then we need to transport them inside the mitochondria. We need to break down these long-chain fatty acids into 2 carbon molecules of acetyl-CoA, and now finally we can enter the Krebs cycle. So as we said, this is not an easy process. As we will see, it's taking quite a bit. So if we need energy very fast, that's not the way to do it. However, once again, if we're exercising after fasting, that's how we make ATP. So we need to use this process. So the first thing we need to do is to mobilize lipids. And basically in this process, triacylglycerols are degraded to fatty acid and glycerol, and they are released especially from the adipose tissue and transported to the tissue that require energy. As we will see now, to transport fatty acids for their usage, we will use a different mechanism. So we won't use lipoproteins, as you will see now. Of course, glycerol is water soluble, so it doesn't really need a transporter. So it can be simply transported in the blood. Now, once, and we will see how they're transported, we need to, once they get to the tissue that needs to use it, they needs to be activated, transported into the mitochondria for degradation, and then, once we get to acetyl-CoA, then we can process them in the citric acid cycle. So, as I said, it's a quite complex mechanism. So, the first step is, it's called lipolysis, and of course, this is what we want to do when we are on a diet, right? We want to mobilize fat from our adipocytes, and we want to do lipolysis. So lipolysis is actually a metabolic pathway through which we start from 3-acetylglycerides and we hydrolyze them in glycerol and 3 fatty acids. And of course, this mechanism, as you may imagine, is induced by several hormones, including glucagon, adrenaline, and the growth hormone, and other that you will see later on when you study physiology. As you can see here from this picture, 3-acetylglycerides will be, will generate glycerol and 3 fatty acids. Now the fatty acid will be used for, beta oxidation and to generate acetyl-CoA. Acetyl-CoA will enter the citric acid cycle, but once again, to start the cycle, you will also need oxaloacetate, and this can be generated also from glycerol. So one thing that we can do is to synthesize from glycerol. However, as you may imagine, when we mobilize lipids, we have a huge amount of acetyl-CoA that is formed. Every 10 carbons, you make 5 acetyl-CoA, right? Every 2 carbons, you make 1. So if you have as I said, if you have a 3-acetylglyceride that has 3 chains of 20, it means that you're making 30 molecules of acetyl-CoA that want to start the citric acid cycle. And on the other side, you have 1 molecule of glycerol. So this is why we really need another source of oxaloacetate. And this is why, and we'll see it at the end of today's class, and this is why, for instance, if we are really starving ourselves, right, if we are not eating anything, and our body will start to mobilize a lot of fatty acids in this way, we will have the big problem is that we have all these fatty acids that get to, the liver. This will actually, of course, potentially have the possibility of generating a lot of ATP, but the liver does not need all that ATP, right? So as we said before, what the liver will do is that on one side, it will generate sugar from gluconeogenesis so that other cells can use beta oxidation to produce ATP. And actually, as you will see today, since really we have a lot of acetyl-CoA that is generated from fatty acid, and we do not have enough, even if we can produce sugar through gluconeogenesis, it's not enough to metabolize them through directly. We convert some of these fatty acids into ketone bodies, and this is how we get rid of some of the carbons, as you will see today, through acetone, so that unfortunately we throw it away, but we need to get rid of it at one point, because there are also issues related with pH alteration, because we are mobilizing all these free fatty acids, and so we cannot really keep them there if we are not using them. So lipolysis is here, of course, a rather simple process. We have lipases that need to hydrolyze these ester bonds in triglycerides. These lipases, they're different, of course, because we start with 3 fatty acids, then we form the acylglycerol, and then we have monoacylglycerol, and finally glycerol. The first rate-limiting step of this lipolysis involves this enzyme ATGL, which actually now we know that is sensitive to hormones. In the past, we were saying that the second enzyme, the one is called HSL, which actually means hormone sensitive lipase, was thought to be the hormone sensitive enzyme activated by hormones, but now we know that actually the first step is also sensitive to hormones and regulates the whole process. Now we have a third one, the monoacyl glycerol lipase, MGL, which will break monoacyl glycerol into a free fatty acid, the third molecule of free fatty acid, and glycerol. So once again, like every biological process that we encounter now, this process is regulated by hormones. As you can see here, we have a very similar mechanism to the ones we have seen before. We have a receptor for these hormones. So glucagon and epinephrine will serve as ligands. Guess what? To bind to a G protein coupled receptor on the surface of the cell, generation of CMP, activation of protein kinases, and so on. We've seen this mechanism before, right? But once again, here, what I want you to focus is on what happens. So we are forming free fatty acid and glycerol. So once again, the mechanism is always the same. The activation of the G protein will activate the adenylate cyclase that will take a molecule of ATP and generate CMP, and this activates the cascade of the protein kinase A, which phosphorylates, in this case, perilipin, which is also known as the lipid droplet associated protein. This actually causes a sort of rearrangement of the lipid droplet, the activation of ATGL, which converts 3-acylglycerol into the acylglycerol and one molecule of fatty acid. Once again, although HSL was thought to be the rate-determining enzyme in lipolysis for some time, now we know that actually the first enzyme is also sensitive to. There is, for instance, this disease that is called the Chanarin-Dorfman disease, which is a condition in which basically fats are stored abnormally in our body, and this is because we have a defect in ATGL, so we are not able to hydrolyze correctly and break down fat, and so they accumulate in organ, including skin, liver, muscle, eyes, and ears, and people have all these defects that they actually usually present at birth. Of course, it's a genetic mutation that is causing this. Now, how do we transport free fatty acids once they're formed? Once again, we have glycerol that is soluble in water, so there's no problem for glycerol. But what happens to fatty acids, right? We had to make glycerols. We make all these lipid particles to transport them. So how come we don't do the same thing for when we use them? Well, for the usage actually of lipids, since they are going the opposite direction, right? We want to use them. We don't want to store them. We need to have a different mechanism. So the most abundant protein in our body, albumin, actually, it binds many proteins, but of course, and it has different, as you will see, that it has different purposes. But the main goal of albumin is actually to transport free fatty acids. So it binds the fatty acids and carry them in the blood. Here, I'm sure that some of you will, might study these things in the future in more detail. The structure of albumin. I'm not going to ask you how the structure changes when it binds the fatty acid. Don't worry about it. I just want you to emphasize that it's the most abundant protein in our blood. We produce about 15 grams of albumin per day. And indeed, in fact, when we need to analyze other proteins that are present in our blood, this is a problem. So when we analyze and we want to check for other proteins in the blood, albumin is covering all of them because it's in very high concentration. So when we do analysis of other proteins in our blood, one thing that we need to do is to remove or try to remove albumin so that we can see the other things. On the other hand, the problem is that a lot of things will stick to albumin. So if we remove albumin, we remove also some other proteins. So it's really difficult to study proteins in the blood. I was telling you one of the first lessons that we do what is called lipidomics, where we try to study all the proteins that are in our blood, and it's actually one of the most difficult techniques that we have right now. Especially because of this very abundant protein like albumin, which tend to cover all the other proteins. So if you think about it, the other proteins are in very small amount. If you're testing for a disease by recognizing the change in concentration of a protein that is present in a small amount, it's very difficult when you have so much albumin. So anyway, fatty acids are transported with albumin. Glycerol is transported because it's soluble, it's absorbed by the liver, and then can have a different fate. One of them, as we already mentioned, is that it can be phosphorylated once it's in the liver, and as you can see here, it's converted into glyceraldehyde 3-phosphate. Where did we see this molecule? Glyceraldehyde 3-phosphate. Where did we see it? In glycolysis and in gluconeogenesis. So if we need ATP, right, we can go on with glycolysis and make use of, or if we need to make glucose, we can also go the opposite way, depends on where we are. Okay. Now, how do we then pick up fatty acids that are bound to albumin and take it inside the cell? This is another topic that has been debated for at least 2 decades to understand really how this process occurs. Of course, if you think about it, fatty acids should be somewhat permeable to the cell membrane because, of course, they have a big chunk that is hydrophobic. On the other hand, if you think about a free acid, a free carboxylic acid, especially under physiological condition, the acid is deprotonated. So if it's deprotonated, it has a negative charge. So if it has a negative charge, it's not that easy to pass the cell membrane that is hydrophobic. Now, as I said, this has been debated for a while. On one side, they found transporter of fatty acids that are present on the cell membrane, so that they found proteins, membrane-associated fatty acid binding proteins that not facilitate the transport. But also, what it was found is that when it gets close to the membrane, there is a slightly change in the local pH, and therefore the fatty acid is not deprotonated, but is protonated. So now it becomes more soluble in the cell membrane, so it can pass the cell membrane. So there is an evidence in support. There are evidences in support of protein-mediated fatty acids uptake, but at the same time, there is this so-called flip-flop mechanism where, as you can see here, free fatty acids get close to the membrane, get protonated, get inserted in the membrane, they flip on the opposite side, they release the proton because they get deprotonated, so now they cannot really stick to the membrane anymore, and so they are released inside. Whatever you want to believe, for sure, both mechanisms are occurring. For sure, we still need to understand fully this mechanism, but at the end of the process, the free fatty acid will enter the cell. So no matter what, this mechanism suggests that the biological membranes are not a barrier for fatty acids. So fatty acids can enter the cell. Of course, from a physiological point of view, it would be desirable to regulate the free fatty acid entering into the cell. Of course, you know, we're introducing a free carboxylic acid, so you know, we should try to limit the uptake when the concentration is high, and we don't need we do not need to transport them. Here's a paper, if some of you wants to read, is you know, quite interesting about this overall transport and the different mechanisms that have been shown over the past years. Now, yes.
Can you explain the flip-flop mechanism again?
Yes.
So co-create.
So to pass the membrane, you need to be very insoluble, right? You need to be you need to fuse with the membrane. So what happens is that you know, free fatty acids on the outer side, when they reach the cell membrane, they get protonated because there's a local change in pH. So they get protonated. Now they can enter the cell membrane, and you know, they can navigate inside the cell membrane. Once they reach, they flip, and they reach the other side. On this other side, there's another change in pH that gets the free acid now deprotonated, so it is negatively charged, so it cannot be inserted, cannot be accommodated anymore. Especially the negative charge is pushed away from the cell membrane, and the molecule will fall out. Of course, now we get to, I think, the most interesting part of the whole mechanism. So how we actually use fatty acids when they're inside the cell. Now, you know, we said it many times when we talked about, you know, how we utilize food, right? So what do we do when we want to take energy out of food? All our catabolic pathways, what do they have all in common? That we do what? We oxidize or reduce food. We oxidize food, right? So we extract energy through electrons, right? So here is the same thing. We have fatty acids, we oxidize them to get energy out of it. So someone. Hello? Okay, so when we do these processes, as you can see from this slide, we will generate reduced equivalents of NADH and FADH. So once again, we will generate ATP by the respiratory chain. Okay, but before getting there, what we need to do is, again, one mechanism that is common that we have seen also in the other metabolic pathways is that to allow the generation of energy, to allow to use this energy that is contained in these molecules, sometimes we need to spend some energy. I need to spend some initial energy to activate the molecules. And the reason, once again, is because we are running some reactions that are not thermodynamically favored unless we destabilize the starting molecule, right? In the first reaction of glycolysis, we need to make glucose 6-phosphate because to drive the rest of the process. To make glucose 6-phosphate, we had to use ATP. And here is a similar idea. So when we had a free fatty acid, as you can see here, we need to use a molecule of acetyl-CoA. We need to attach the coenzyme A. So be careful, I'm not going to ask you about the structures, but as you can see here, we start from a molecule of fatty acid, right? And we exchange the coenzyme A with the acetyl group. Okay, now what you form, basically you kick out the acetyl group, basically you kick out the acetate, which is a carboxylic acid with 2 carbons, and you attach the acyl groups, which is the long chain carboxylic acid of the fatty acid, right? So what you do in the first reaction, you activate, once again, we have seen this before, we activate the molecule so it's ready for the next step by binding it to coenzyme A. Who remembers what kind of bond is that with coenzyme A? It's a thioester, right? The coenzyme A ends with SH and it's able to do, with the free acid, we can synthesize a thioester. Okay, so once again, this makes the carboxylic acid more reactive for the further utilization, and once again, this of course requires energy. So actually, this is a 2-step reaction that requires ATP. So first of all, we react the fatty acid with ATP, we form acyl AMP, and then we react it with coenzyme A, and we form the thioester that your friend was referring to. Now, once we have the acyl-CoA inside the cell, we need to reach the mitochondria. And of course, acyl-CoA is not able to get to the mitochondrion unless we transport the molecule in the mitochondrion. And how do we do this? We do it through a mechanism that involves, once again, some critical enzymes, critical proteins, and in this case, a transporter, which is called carnitine. So what happens here is that once we synthesize the acyl-CoA to make it permeable to the outer mitochondrial membrane, we need to transfer it from the outside of the mitochondria through the outer mitochondrial membrane, the inner mitochondrial membrane, and then finally use it, as we will see, to make molecules of acetyl-CoA through beta oxidation. The molecule that we use to shuttle the carboxylic acid is called carnitine. Has anybody heard about this molecule, carnitine? Why? What are the bodybuilders in this room?
They got flexible.
Yeah, so bodybuilders, when they want to burn fat, or even if you're not a bodybuilder and you want to burn fat, one of the supplements that is most sold on Amazon, right, is acetylcarnitine. Why do people take acetylcarnitine? Because they hope that they will increase, right, the amount of carnitine that they have inside their mitochondria. Because we need to transfer, once again, the fatty acids to burn fat. We need to transfer it to the inside of the mitochondrion, and the transporter, as we will see now, it's this molecule, carnitine. So we actually form acylcarnitine to transport it inside, to transport the acyl group inside the mitochondrion. So this mechanism is actually quite interesting. So we have the fatty acid that is inside the cytosol that has been activated with the coenzyme A, And as you can see here, what we do is basically we release the coenzyme A that was only used to activate the free fatty acid, and now we bind the fatty acid to carnitine. And now carnitine that forms acyl carnitine. So basically, as you can see here, there is this R can be, any length. It depends on the fatty acid. The fatty acid is now bound to a carnitine, and we will see now what is carnitine, and it's transferred inside mitochondria.
We don't see.
Oh, you don't see the pointer, but this is, I seriously don't understand why they have this system. So I don't see the pointer on my screen, and it's on yours, and not vice versa. So, okay. So as you can see here, now with the pointer, you have the acyl that is bound to the coenzyme A. So the fatty acid is bound to the coenzyme A and is activated. Now the coenzyme A is released, and then we attach the fatty acid to carnitine, and then we transport it inside the mitochondria. So in fact, the reason why, if you look on Amazon or any other sources of carnitine, they can sell it as a free carnitine or as an acetyl carnitine, and people tend to buy acetyl carnitine, and it makes sense. Because carnitine itself is quite polar. So if you really want to increase the amount of carnitine inside the cell, you should find a way so that it can actually enter the cell. So by introducing acetyl carnitine, so a carnitine that is made more hydrophobic, it's easier to make it enter the cell. Of course, there is not real proof that by eating carnitine, you do increase the amount of carnitine inside your cell, or at least it does increase. How much it does, what is the bioavailability of the molecule is not completely sure, and so every company can claim that their carnitine is better at doing that, and they can sell it at any price, and people will buy it, right? Of course, the end goal is to have a higher concentration of this transport inside our cells so that we can actually use fatty acid more efficiently. And this is why, as someone mentioned, right, when they want to, bodybuilders want to burn fat before they exercise, they usually take carnitine. But as you will see, now, many supplements that are given especially to older people or people that suffer with neurological disorders, they do contain carnitine because it really helps in the metabolism of fatty acids, and especially when you get older, it's really important to have this pathway very, very, very active. So once again, before acid-CoA can be transferred, it needs to be converted into acyl carnitine. Carnitine is essentially made of 2 amino acids. As you can see here, it's actually biosynthesized from this compound and 3-methyl lysine. In humans and in many other animals, actually, carnitine is either obtained by the diet, so we eat it because we are eating food that contains carnitine, because the cell of the animal that we are eating contains carnitine, or is biosynthesized from TML. TML is a non-protein amino acid. The origin is not completely understood today. It's generally accepted that TML basically derives from the hydrolysis of proteins that have been post-translationally modified, and so this is basically a derivative of lysine after post-translational modification, and so once again, it's critical. The intake of carnitine to activate this process. You know, from a clinical point of view, the inability to synthesize carnitine actually it has been recognized as a contributing factor to the development of autism in males, and there are several, you know, symptoms of carnitine deficiency in several organs, and you know, muscle weakness during prolonged exercise is a clear symptom of deficiency of carnitine, and this is again why athletes tend to supplement with carnitine. Now, in the final part, I promise, of today's class, I see that you are really tired. 4 hours in a row are quite difficult. Is fatty acid beta oxidation. So in the last part of today's class, we will see how we do actually synthesize acetyl-CoA from fatty acids. Now, of course, someone would, you know, already tell me that, you know, fatty acids are not all the same. There are even and odd fatty acids. Some contain double bonds, some contain multiple double bonds. So what we see here as a general process is the process of metabolizing fatty acids that are, you know, with an even number of carbons, so that we can easily, you know, count the number of molecules of the acetyl-CoA that we make. We just divide by 2 the number of carbons of the fatty acid. But we will see also what we do when we have an odd number, or when we have double bonds that are present. Now, beta oxidation is easier than the other processes that we have seen before, because it's just 4 reactions. So once the fatty acids are inside the mitochondrial matrix, they get oxidized by beta oxidation. You will understand now, because you know the reason of the name. And basically, this process occurs with a sequence of 4 reactions. What happens at the end, and I think, you know, what will stick in your mind after you're done with this course, and you know, with biochemistry, what you should stick in your mind, that every time we run one cycle of beta oxidation, what we do is we remove 1 molecule of acetyl-CoA from fatty acid. So if a fatty acid is 20 carbons, we run 1 cycle of these 4 reactions, and basically what we get is 1 molecule of acetyl-CoA, and then we get a molecule of fatty acid that is 2 carbons shorter. So if we have 10 carbons, how many rounds of beta oxidation do we have to do? Four, okay, 4, because of course in the last round of beta oxidation we have 1 molecule with 4 carbons, right? If we split that in 2, if we remove 1 molecule of acetyl-CoA, it remains 2 carbons. That is already a molecule of acetyl-CoA, as we will see. So please do not get tricked. This is one question that I always ask, that I've asked at the exam, right? How many cycles of beta oxidation you need to completely oxidized fatty acid that is of 20 carbons, right? One of the answer would be 10. Of course, that's wrong, right? We need 1 less than the number of molecule of acetyl-CoA that you get. Do you understand why? If you don't understand it, we'll see it now. So in the first step, we are doing an oxidation. As you can see here, once again, if we are oxidizing a compound, we need a coenzyme that gets reduced. It's FAD that gets reduced to FADH2. And what we do here by this oxidation is basically we are forming a double bond by removing 2 hydrogens that are next to in the alpha position of the carbonyl group. Now, the second step is an hydration reaction. So we basically add a molecule of water, we form an alcohol. And as you can see here, it's a secondary alcohol that can get oxidized in this 3rd reaction to a ketone. And now, as you can see from the final step, thiolysis, what we do is we basically are breaking down the molecule where the ketone was formed. In this reaction, we have a thiolysis, and we form an acyl-CoA, and we detached a molecule of acetyl-CoA. So in these 4 steps, basically, we are cutting off a molecule of acetyl-CoA, and we are generating one molecule of acyl-CoA with what is shorter of these 2 carbons, right? So this process needs to be repeated over and over, and to get to the acetyl-CoA. So I have a question. What do you see from this process? What do we need to run this process besides the fatty acid? Coenzyme in what form? Oxidized, right? So once again, this process cannot occur unless we are able to reoxidize these coenzymes. So once again, if you want to burn fat, you cannot do anaerobic exercise. You need to do aerobic exercise because you need to have oxygen to start, breaking down with beta oxidation. So I don't have, time to go over the reaction one by one, but I already did more or less. I think that it's quite simple. Once again, oxidation, hydration from the alcohol gets oxidized to a ketone. This beta keto acid can be hydrolyzed, and the final reaction gives 2 fragments, 1 molecule of acetyl-CoA and 1 molecule of acyl-CoA. So the new fatty acid, that is 2 carbons shorter, is already activated with the molecule of coenzyme A, and so it's ready for the next step. Is it clear? Are you still awake?
Is this the same where we have a fatty acid that has double bond?
No. Wait. Well, now that you asked the question, what do you notice in this molecule that we form after the second step, after the first step? What kind of double bonds does it have? Trans, right? What did we say about fatty acids that we normally eat, that they normally have double bonds? Are they cis or trans? Hopefully they're cis, right? We said the trans are not very good. So actually, the problem is that, someone would say, well, you just cut down the fatty acid, and then when you get to the double bond, you're already there, right? You don't need to do the first reaction. But as we will see now, the problem is that we need to we need to have a double bond. And of course, if the double bond is already there, it's fine. But the problem is that the stereochemistry of that double bond is not the correct one most of the time, because we have a cis double bond and we need to get a trans double bond. Guess what we're going to do? We're going to make it, right? Become trans. So, here again, just to give you an idea. So if we do an oxidation of palmitate, the degradation of palmitoyl-CoA requires 7 cycles to reduce the fatty acid to 8 molecules of acetyl-CoA, generating 7 NADH and 7 FADH2. And this means that we can generate ATP by reoxidizing these coenzymes. Now, not only we directly generate reduced coenzymes, but remember that every time we do one cycle, we also generate 1 molecule of acetyl-CoA, and this acetyl-CoA can enter the Krebs cycle. So the oxidation of palmityl-CoA generates the 108 ATP molecules, which is quite a number if you think about it. Now, of course, the activation of palmitate consumes 2 ATP molecules. Once again, who cares, because we're making way more ATP molecules than what we need to use to activate the molecule. So what happens if we have an odd-chain fatty acid? You cut. At one point, you get to a molecule that has 5, right? 5 carbons, right? Because you remove it 2. So what basically happens is that at the end, when you have an odd number of carbons, at the end, you will have 2 molecules, one of acetyl-CoA, which is normal, but the other one has 3 carbons instead of 2. So it's a molecule of propenyl-CoA. No, so now propionyl-CoAs is actually ultimately transformed to succinyl-CoA through these reactions. I'm not going to ask you to remember these steps, but but what happens to succinyl-CoA, maybe you remember, right? Succinyl-CoA can enter, right? Succinyl can enter the Krebs cycle. So this is a way of re-entering the Krebs cycle. And actually, some of the intermediates that we can make of the Krebs cycle can also be used for gluconeogenesis if it's needed sometimes. So what happens once again when we have unsaturated fatty acids? When we have unsaturated fatty acids, the problem again is that when we shorten the chain through beta oxidation, we get to the point where we have the double bond that is in the wrong configuration because it normally cis. So basically what we do, we get to the trans, we have an isomerase that will isomerase the cis double bond from the trans double bond, and then the beta oxidation can proceed as usual. So clear? So no problem at oxidation an odd number, no problem in oxidating the presence of double bonds. And of course, if we have the oxidation of polyunsaturated fats, it requires some additional enzymes that will catalyze the reduction of the double bond and the formation of a single bond, as you can see here. But again, these are really very peculiar pathways. Don't worry about them. I'm not going to ask you in the exam. But one thing that I already mentioned, I think is quite interesting, is that fatty acid degradation occurs primarily via the beta oxidation in mitochondria, but actually that's not completely true because beta oxidation can also occur in peroxisomes, and if you remember, peroxisome are these organelles that are bound to the membrane, and usually the first part of the beta oxidation of long chain fatty acids. So when we have fatty acids that usually exceed 20 carbon atoms, which is common, they get first beta oxidized in the peroxisomes and then shuttled for further oxidation in the mitochondrion. And this is also important because peroxisomes also are used to synthesize some important fatty acids, with long chain fatty acids. So this is how also we can synthesize them by shortening longer ones. There are, of course, diseases that are associated to defects in this process. The Zellweger syndromes are, again, genetic abnormalities that involve problems in breaking down long chain fatty acids, and in particular, patients affected by these pathologies have mutations in peroxins. There are proteins that are that help forming peroxisome, and basically what happens is that long chain fatty acids cannot be broken down and they start to accumulate, and of course this is a problem because we cannot use them and they get accumulated, and this will also cause also big problems because we cannot synthesize some important fatty acids that are synthesized in peroxisome that are also specifically very important for neurons, and these patients have complication, neurologic complication, and also complication in the muscle and the liver, and so on. So in the very last part of today's class, I want to just briefly touch on ketogenesis. What is ketogenesis and why it's important and why it's so popular? It's been so popular now, in many diets. So, what is ketogenesis? Ketogenesis is a process by which we generate, these compounds that are called ketone bodies, and they are originated basically from the acetyl-CoA that we get from fatty acids. So let's try to understand, when we have ketogenesis and what is the purpose of this mechanism. So if you think about it, if you are in a prolonged fasting state, right? You're doing an extremely low carb diet, or not only that, but you are introducing a lot of fat with your diet. So there is this ketogenic diet where you eat a lot of fat. So for sure, you will have a lot of fat that gets to your liver, right? Now, if everything works fine, this is fine. But remember that this is not fine most of the time, because especially if you're not that young, all the processes that we have seen now, they're not working that well. Now, of course, there are some advantages to this diet. You can get lean very fast, but there are also some problems. So anyway, if our carbohydrates are unavailable because we use them up, what will happen is that fat cells will mobilize fatty acids. Fatty acid will get to the liver, and once they get to the liver, we have once again a problem because we do not have enough glucose to use them. Moreover, when you are in a fasting state, as we mentioned many times, the tissue that is suffering more is our nervous system, because we normally use glucose as an energy source. So now what the liver does is trying to help our nervous system to survive, to produce ATP through a metabolic switch towards the OXPHOS. Now, to run the Krebs cycle, once again, you need acetyl-CoA, and you need glucose, you need oxaloacetate. So what happens here is that the liver, instead of using the acetyl-CoA that is generated from fatty acids, it will convert it into molecules that can be transported into the blood and can be picked up by the brain, the neural cells, and at the same time, it will generate through gluconeogenesis the glucose that is needed to run the Krebs cycle. So in other words, as we mentioned already before, the gluconeogenic path that is activated in the fasting state by the liver is not done by the liver for the liver. The liver will release the sugar in the bloodstream, and this will be picked up by the brain that has a metabolic switch and will use ketone bodies. So where are ketone bodies produced? Well, they are produced mainly in the mitochondria of the liver cells, and of course, their synthesis will occur in response of the unavailability of glucose, such as during fast. Now, of course, other cells like human astrocytes are capable of carrying out ketogenesis, but they are not as effective as in the liver. What I want to tell you is that ketogenesis occurs constantly in our body. So it's not that we do only activate it under these circumstances. So why it's also important, this path when we want to lose weight, because of course activation of ketogenesis in the liver at the same time will inhibit lipogenesis, promotes fatty acid oxidation, and of course switch off acetyl-CoA carboxylase, as you will see later on. That is done when we do the synthesis of fatty acids. So ketogenesis in general happens when we have low glucose levels in the blood, and it can also happen when you have situation like type 1 diabetes, when you have a lack of insulin. Of course, this will reduce the possibility of utilizing glucose in our blood, and therefore our cells will suffer, and we'll have to use this pathway to generate ketone bodies. So here are the 3 molecules that we generate from acetyl-CoA. So as you can see here, ketone bodies biosynthesis is basically a condensation of molecule of acetyl-CoA. So you have to understand, once again, the setting. We are starving, we mobilize a lot of lipids. These free fatty acids get to the liver. The liver doesn't know what to do. So what it does, it will break them down, form molecules of acetyl-CoA. This molecule of acetyl-CoA will generate these ketone bodies, and most importantly, they will release the coenzyme A. Because, the problem, as someone would ask, okay, but why doesn't, the liver simply generate acetyl-CoA and then release acetyl-CoA? But the problem is that acetyl-CoA does have the coenzyme A, but we want to recycle this coenzyme A. So by forming the ketone bodies, we release coenzyme A, and coenzyme A can be used for other cycles of beta oxidation. So we can continuously beta oxidize the lipids, we cut them down, we form, these ketone bodies. Some of them are, released. We cannot use all these ketone bodies. Acetone is released through breathing, right? If somebody is doing this kind of diet and reads the book, they tell you to eat, mint, or that they tell you, don't kiss your boyfriend or girlfriend for a while because you're going to have bad breath, and the reason is because you are activating ketogenesis. So they suggest you to eat, candies with mint or things like this, and the reason is this, not because they're making you leaner. It's just because you're going to have bad breath. But besides acetone that is, is released, then the other ketone bodies can be used, and as we will see, we can basically convert them back into acetyl-CoA, and then acetyl-CoA can be used for the Krebs cycle. So in other words, the liver makes glucose, ketone bodies, send them to the brain, and the brain picks them up and use them for ATP production. We asked several times this question: can we make glucose from fatty acids? And as we said, the general answer is no. And of course, we're going to have a question at the exam, right? Nobody's going to make a mistake on that. Why can't we make glucose from fatty acid? Yeah, because how is it called, this reaction? Pyruvate decarboxylation, oxidative decarboxylation is irreversible, right? We cannot generate pyruvate from acetyl-CoA directly, but it has been shown using large-scale analysis in the cells that we do actually have the possibility of generating glucose from acetyl-CoA here is the scheme. I just want to tell you that from an energetic point of view, there's no advantage. It would require too much, and it does require too much energy to generate glucose. So it's not. I mean, although they've found enzymes, they are doing this in the cell. This process is not really active. So ketone bodies are actually used in other tissue, from the liver during fasting. Some cells actually prefer to use ketone bodies, like the heart or the renal cortex cells, over glucose. The brain prefers glucose, as we said, but beta-hydroxybutyrate and acetoacetate, there are the two ketone bodies, can pass through the membranes easily, and therefore they are source of energy when we cannot metabolize sugar, or we don't have enough sugars. So actually, believe it or not, when we are under this condition, so if we are starving, actually the brain can get up to 70% of its energy through ketone bodies. And of course, part of the reason why, people that are in favor of this diet are saying that they're good is, once again, because we also activate gluconeogenesis, that, as you will see next week with Professor Graziani, will start by degrading proteins that are misfolded. So they have shown that this process activates autophagy and activates all these, sort of recycling processes in our cells, which, are considered to be good. Once again, as you will see, there are also the bad sides, that you will need to get rid of all the nitrogen that you need to remove from amino acids to generate glucose from amino acids. So once again, ketone bodies can be converted back to molecules of acetyl-CoA, and of course, this is basically, once again, the entrance of the Krebs cycle. Once again, we can generate intermediates of the Krebs cycle also from amino acids, and this will generate oxaloacetate that is needed. But as you will see later on, amino acids can be also used to generate acetyl-CoA and ketone bodies. Yes. Sorry, no, I cannot hear you. Yes.
How can we go back to
As I said, the liver makes glucose and ketone bodies and shifts it to the blood, right? And now the brain picks up both glucose and ketone bodies. Ketone bodies generate acetyl-CoA. From the glucose, the small amount of glucose that is generated by the liver, we actually can generate oxaloacetate. So we don't really use it for ATP production, but we generate a few molecules of acetyl-CoA that will start the cycle.
So in regular acid cycle, we usually go to the acetyl-CoA and the oxaloacetate, but here they have two different words.
Yes. So the question is, do we normally run the Krebs cycle doing this process? No, actually not, because I mean, under normal start condition, of course we are still using fatty acids. So right now I'm using not only glucose, but I'm using also fatty acid. So under these circumstances, I don't need to make much ketone bodies. So in this case, I will make acetyl-CoA mainly from fatty acids, and oxaloacetate mainly from glucose or glycogen, I would say, because I'm not eating. But once again, this slide is probably my final slide. I'm just showing that you can actually do the same thing starting from amino acids. And once again, when we are under, ketogenic diet, where we basically are removing we're reducing carbohydrates intake, we will have to make these intermediates through amino acids. So actually, one thing that might happen, as you will see when you will study amino acid metabolism, and we'll see these extreme situations. What can also happen is that, as you can see here, we can enter the Krebs cycle through these processes where we, transaminated, we convert the Krebs cycle, the amino acids in the Krebs cycle intermediate, but we can do also the opposite, go in the opposite direction. So whenever we are under these stress conditions where we might need to actually use some of the intermediates of the Krebs cycle to generate other important molecules, including glucose, what will happen is that some cells will really suffer, because you will basically, use the intermediates of the Krebs cycle to synthesize maybe amino acids, because you needed maybe to synthesize pyruvate and do gluconeogenesis, and basically you don't have the Krebs cycle working effectively anymore, and you don't have energy in the cell. So once again, these are all mechanisms that, of course, they are physiological. Of course, the nature designed them to work all the time, but it's when you do it, a metabolic switch, which really requires, a heavy use of body starting material by breaking down our internal molecules, then you can encounter a big problem. So I hope that I've convinced you that this is a big problem. Anyway, so I think that this is the last slide of today's class. Of course, the most common disease associated with ketone bodies is diabetic ketosis. That is typical of patients with insulin-dependent diabetes mellitus. So if insulin is not produced and the liver cannot absorb glucose, they cannot provide oxalate to produce fatty acid derived acetyl-CoA. And of course, the result is severe acidosis. Unfortunately, I'm not going to be able to talk to you about the biosynthesis of fatty acids. We already, did together a general idea on how we synthesize them. Of course, the origin, once again, is acetyl-CoA that is generated from an excess of carbohydrates. You will see it next week, and then you will see the rest of the course. So since it's the last actually lesson together, I hope that you enjoyed this course. I know that biochemistry as one of the first courses is not that easy, but I hope that you're finally seeing something that tells you where you're gonna be working on, all these metabolic disorders, metabolic diseases, and many of the diseases that I cited that are very common, heart diseases, diabetes, they're all related to metabolic problems. So I hope that, when you prepare this exam, you really feel that you're not wasting your time, but you're actually putting the main pieces together, so that when you start to study a disease, you understand actually what's going on, or you understand why some drugs that you're prescribing are having an effect. And please, remember my first lesson. Medicine is changing over the past years. I've been attending conferences lately where they only talk about omics, and how by analyzing the changes at the molecular level, we are learning that diseases that we always classified and treated in one way are completely different diseases. So I really think that you will see a big change in medicine in the next few years, and you probably will study new diseases that haven't been studied in the past 40 years or so. So I think that you should be very focused on the molecular basis of the diseases and understanding in the future. And of course, as I said, my lab is open if you want to join now or later on for your thesis, and you know how to reach me. And so if you need me for any help or reason, I'm there. And of course, before the exam, if you have any problems, please don't hesitate to contact me. And of course, good luck. E adesso buon aperitivo.
Grazie mille. La ringrazio anche tanto, Angela, è andato molto bene. Mi perdoni ancora per averla interrotta.